Time to learn of smell and taste.
Hello! This lecture is about two topics. The first are chemical senses, and then the second half will be about senses that humans don't have, but animals do, about all the interesting stuff. But let's start with chemical. The first sense is gustation. What is gustation? This is a fancy word. Just everything related to taste. Good. Next question then. Where are the receptors for taste in humans? And also, what does the word receptor mean here? What do you think? Where are the receptors? Probably in the tongue. It turns out that not only in the tongue, we also have a little bit of those in other parts of our body, including deep in the throat and almost in the border of esophagus. I'm not sure how important they are in normal life. We don't usually realize that we can taste with the inside of our throat. Apparently we can. What does the word receptor mean here? So that's an interesting conversation, and actually I realize now that I totally skipped it in the previous lecture, even though it was relevant in the previous lecture. The word receptor is weird because it can mean three different things depending on the context. And these things are not entirely different, they are closely related. The three meanings are essentially the organ that we use to sense something, so the entire big thing that you could conceivably take in your hands and manipulate. A cell, potentially in this organ, that does the main job of sensing. And finally the protein that sits inside the cell, and that's the context in which we used it when we talked about synapses. A protein that usually sits on the membrane, although not always, and that when activated changes something about how the cell acts. So let me briefly illustrate it here. For example, in case of an eye, and we haven't talked about eyes yet, but you probably heard of those, we have them. In case of an eye, the eye itself may be considered a receptor. That's a visual receptor. But then within the eye, there are special cells that have a certain shape that actually do the job of detecting the light, and we'll talk about these cells later. And then within each cell, if we look deep in, there will be some proteins. I don't know how to draw it, so I'll draw something random. But there are some proteins that actually interact with the light and make sure the cell responds to it. So all three of these levels of organization, all three of these objects at different scales, from something that you can touch to something that you can work with a microscope, and down to something that you can only work with biochemically, all three items would be called receptors, which may be confusing sometimes. And the same is true for this lecture. For example, for gustation, for taste, we will have the organ of taste, what we use, humans, to taste things, and in this case it's probably tongue. So let me draw it roughly. This will be the tongue, if you don't mind. But also in the tongue there will be individual cells somewhere. We will discuss in a second where they sit, but there will be individual cells that will somehow talk to neural cells, and neural cells will somehow send information to the brain. And then within each cell, supposedly, there will be some proteins on the membrane that are exposed to the external environment, in this case the saliva, and catch chemicals from there, right? Because ultimately taste is about sampling chemically what is in our mouth and responding to certain chemicals like sugar. We taste it. All three of these levels, from the organ that's called the tongue and down to special proteins that sit on the membrane. That's supposed to be a membrane, and this is supposed to be a receptor as a protein. All three levels are called receptors. You need to look for context when you're trying to analyze scientific writing, for example. In this case, what does the receptor mean? I asked where the receptors are, which means that probably I was talking about at the level of cells, and I asked in what organs these cells are sitting. And so the answer was they're in the tongue, but also deeper in our throat, although probably there are fewer cells there. Most are probably in the tongue. Next point, how many types of receptors do we have for taste? And in this case, receptors are actually somewhere in between cells and proteins, because here the question is essentially how many different tastes do we have? How many primary tastes do we have? When our receptors detect something, how many types of somethings can we detect? And this is a tiny question, but a very reasonable one, so let's make it the first question where you pause the video, think, give an answer, and then continue watching. Okay, what's the answer? The answer is 5. For a while people thought that there are only 4, and we have normal words in different languages only for 4, because somehow we totally missed the 5th one until mid-20th century. So the classic ones are sweet, sour, salty, and bitter. But then it turns out we have the fifth one, for which we use the Japanese word umami, because Oriental cuisine was a little bit better with catering to this taste. And what are these tastes? Do you know? What is sweet? Mostly sugars, right? Sour is about acids, salty is about salt, bitter is a complicated one. Lots of things are bitter, and we'll talk in a second about why. And what is umami? The classic chemical that we use that tastes of umami is glutamate. Exactly same glutamate that we talked about as a neurotransmitter. The same thing that we use in our brains for the neurons to talk is a basic amino acid, and every time you cook, depending on what you cook, you have more of that or less of that, and you can also just add it. And as I mentioned in one of the previous lectures, there are no scientific reasons to be afraid of MSG, monosodium glutamate. Next, are different receptors all in the same place, or are they distributed across different parts of the tongue? That's a very interesting question, because it is in a way a meme, and not just one meme, but two memes that are sitting inside each other like a Russian doll. You have probably seen this picture. It used to be in all textbooks, but you have probably also heard that it turned out to be a lie. So the interesting part that the statement that it is a lie is also itself a lie. Here's what happened, because it's kind of funny. In about 1901, a person called Dirk Henning, who back then was in Germany, studied whether different parts of the tongue have different sensitivities for basic taste. And I'm guessing back then there were 4 basic tastes, because I don't think he studied umami. And he figured out that actually different parts of the tongue are sensitive slightly differently to different tastes. There are parts of the tongue that seem to be more sensitive to bitterness, and there are some parts that seem to be more sensitive to sourness. And then he published it, and everybody were like, oh, fun, that's nice, different parts of the tongue for different tastes. That sounds like a theory, that sounds like an interesting story. So they started to include it in textbooks, and then one textbook would reprint another textbook's claims, and gradually these claims would get amplified. The original paper actually only claimed that the sensitivity is slightly different, but by about mid-20th century, textbooks started to claim that different parts are responsible for different tastes. You can see how it's a slippery slope. When different parts of the tongue have different sensitivity, it sounds reasonable to say that different parts of the tongue are primarily responsible for detecting different tastes. But then the word primarily may slip, and you have only responsible, and then somebody who forgot the original claim would have read the word responsible and think, well, what does it mean responsible? It means that it tastes it, which kind of suggests that maybe other parts don't. And so the claim became stronger than it was originally intended to. Then in 1974, a researcher called Virginia Collins decided to test that. She wrote a paper, Human Taste Response as a Function of Position of Stimulation on the Tongue, and also Soft Palate, which is like up there. And basically she figured that the original statements of Dirk Henning stood. There were some minor changes, but she found that different parts of the tongue were differently sensitive to different tastes. But again, it doesn't mean that the part that can taste sourness a little bit better cannot taste sweetness. It can, it just tastes sourness in a slightly lower concentration. So if you're drinking something that's very mildly sour, the sides of your tongue will notice the presence of this taste earlier. But eventually, if you're drinking juice, obviously your entire tongue will be involved. And if you want to imagine this difference in sensitivity, imagine that you got juice and you diluted it with water, like a lot, so you still have taste but barely, and then you dilute it 3 times more. Now this barely flavored water, when you drink it, may activate one part of your tongue, but not the other. So it's really a subtle point. But the interesting situation is that Virginia Collins essentially confirmed the original study, but now her study is cited as a disproval of the original study because people kind of forgot what the original study was. And I guess it's just really interesting for people to think that there was a meme in the textbook and that textbooks were wrong for so long. And now this had become a meme in itself. So the truth is that different parts of the tongue are sensitive differently, but the differences are very tiny, and that's funny. Next, why do we have tastes at all? What's the point in feeling tastes? I have kind of answered this question by now. We want to know the chemical constitution of what's in our mouth. It's a good idea to know what's in our mouth, but we don't need to know that in detail. It's enough to have some basic idea of whether it's edible or not, and what kind of food is it, probably. So let's think about it. I'm not actually sure what is sourness. I mean, why would we care? Why would animals care about the amount of acid in our food? I'm not sure how it is helpful, but you can definitely figure out ecologically, evolutionary, why there is a point, why it can be a beneficial trait to know the amount of sugar in your food, to know the amount of bitterness, the amount of umami. Sweet, bitter, and umami. Maybe salt as well. Why is it evolutionarily beneficial to taste them? To be able to taste them? Think about it. Pause the video and think about it. Alright, what is sweet? Sweet is sugar. Why do we care? Sugar is essentially high energy food. Our ancestors ate fruit sometimes, and it's nice to have sugars. You need to know a little bit of chemistry to appreciate this idea fully, but sugars is what we use for energy. We break them down biochemically inside our cells. If you eat sugar, you have access to energy in just a matter of few minutes. If you need to run somewhere, eating a sugary thing would give you this boost of energy in a short amount of time. And so that's probably why most people or many people have a sweet tooth, because it was a rare treat back in the day when sugar wasn't that easily available. It was like some sort of a super boost in a video game or an energy drink, something like that. It's something that gives you an unfair advantage over a course of an hour and then you crash. Then salty things are important because we use salt a lot in our body. Remember all these ions, sodium and chloride? If you eat grass, for example, grass is not particularly salty. It has some salt because it's also alive, so it has to have sodium and chloride in it, but supplies of salt that it provides are pretty meager. At the same time, as an animal, you need lots of ions in your body. So if you want to make friends with a deer, or a moose, you actually don't want to give it sugar, you want to give it salt. They will be really grateful because they need it as much as we do, but they don't have access to good sources of salt, eating grass and twigs and branches, for God's sake. And umami, glutamate, is essentially meat. Meat is rich with proteins. Proteins are made of amino acids, and when they break down, for example, if you cook food, these amino acids get released and we can detect them as umami taste. So in a way, the fact that we can taste sweet means that our ancestors used to eat fruits. And the fact that we can taste umami shows that our ancestors used to eat meat as well, which kind of betrays our omnivorous origins. But the most interesting taste of all is actually bitter, because for all other tastes, we can pretty much name the chemical that it is about. Sweet is about sugars, and there are several sugars, and then there are also some molecules that pretend to be sugars, but basically that's a very narrow group of chemicals. Salty is salt, which is pretty much sodium and chloride. Both independently are salty, and the combination of both is the saltiest. Umami is meat, amino acids. There are some other chemicals that also activate this receptor, but incidentally, they also come from meat. But for bitter, there are hundreds of different chemicals that are bitter to us. So this experience of all these chemicals is glued into one impression, the impression of bitterness. Why? Because they're bad. Basically everything that's toxic. Think about it. If we identify it in the food, we may survive. So having a mutation that makes a poisonous thing bitter would save you because you would spit the food out and you wouldn't die, while some other animal of your same species that cannot taste this chemical may suffer and have fewer babies, and thus have fewer of its genes remaining in the population. And that's interesting because by noticing things that are bitter, we can figure out what is or used to be hurtful for us. And we can also figure out which chemicals our ancestors met, because we would not be prepared for the chemical that our ancestors have never experienced. Evolution cannot think ahead. And this means that from this list, we can make connections to two previous lectures, because bitterness, the idea that some tastes just warn us about danger, is kind of a little bit like pain. It has the same undertone to it. It's an indication to the body that something is happening that perhaps shouldn't be happening. Even though we actually learn to eat some bitter compounds and sometimes even enjoy it, like think of mustard or horseradish or something like that, which are hot but also bitter. And the situation with sweets that we crave resembles another phenomenon that we discussed before, when something is originally good, but then we find an artificial substitution that feels even better, even though it's no longer helpful to us. Remember that? It's kind of like a supernormal stimulus. Our ancestors were really happy to find the ripened mango, and they would eat it, and it would give them an extra boost to catch another squirrel. But now we can just buy a bag of sugar and a spoon and sit there eating sugar. Nobody eats sugar like that, I guess, but people totally eat Nutella like that. And that's no longer helpful. Our teeth suffer. Okay, so this was some interesting logic, but it was also a little bit of hand waving. It felt like we cannot really test many of the statements because we don't have access to the past. We don't have direct access to our ancestors. We're making some guesses about our ancestors based on common sense and these facts, but can we actually test that? Probably not. What we can, though, we can look at other animals and try to see whether there is any rhyme and reason here. So let's look at a Drosophila for a change. Remember those little fruit flies? What do you think? Where would a Drosophila have its taste receptors? That's the first interesting question. And the second interesting question, what tastes will it probably have? And also, what chemicals will be bitter to it? So again, the questions are, imagine the fruit fly. Where are the receptors? What are they sensitive for? And which chemicals are bitter? Pause the video, think about it, then compare your answers to the answers given here. Let's see. Let's start with the receptors. Drosophila, as you probably know, if you've ever had them in your room and if you ever had a half-eaten apple forgotten on the countertop, they reproduce in rotting fruits. They lay their eggs there, and then little larvae grow and they eat this rotten fruit, and then you have more fruit flies, which means that sweet would be the key taste for them, right? Because they need to identify fruits that are ripe, at least ripe enough for the larvae to survive in them. Then, of course, they will have bitterness in case the fruit is actually poisonous or something, in case it has chemicals that are not helpful for the Drosophila. They also have a water receptor, and we also have a water receptor. We just cannot consciously perceive it because, we don't feel anything when water is not present. And then they have high salt and low salt. This means that there are two proteins and two types of sensory cells that respond to different concentrations of salt. I am not sure what it means in practice and how to relate to that. We don't see a receptor for umami here, which kind of makes sense because meat doesn't mean anything for a Drosophila. We also don't see a receptor for sourness, but maybe it's tasted differently. Let's park that. The interesting part is where the receptors are. Okay, if you were a fruit fly and you would lay your eggs on a rotten fruit and you would eat rotten fruit as well, where would you want your receptors to be? Now, the world of a Drosophila is very different from a human world. So the best place for receptor in this case is actually your legs and maybe wings and the proboscis, which is what you eat with. But legs are your best bet because you're so tiny that you basically land on food. That's very different from how we eat, right? When we eat, we typically take food with our hands and bring it to our mouth because food is much smaller than us. But for a Drosophila, a typical size of a food item is kind of the size of a small city for a human. So if our food was like that, we would have probably also tasted it with our feet, wouldn't we? Ethanol is bitter to flies. Ethanol is not bitter to humans, but it's bitter to flies. Why is it bitter to flies? Can we come up with some evolutionary explanation for that? Does it make sense? Yeah, it totally does, right? Because where does ethanol comes from? It comes from rotting fruits, which means that the same very thing that you seek, that you eat and that you raise your young in can also kill you, depending on how far it is in the fermentation process. So if you're a Drosophila, you should be very careful. Drosophila are actually attracted to the scent of ethanol, which is not taste, it's smell. We haven't talked about smell yet, but if you ever left some alcohol out, you would know that Drosophila are really attracted to it. But at the same time, ethanol is bitter to them, so they have this love-hate relationship with ethanol. One would say they're attracted to it, but they also hate it, because if you bask in it and you put your babies in it, it would kill you and your babies, right? Depending on the animal, the same chemical may be sweet to some, neutral to others, and bitter to others. Saccharin, which is sweet to us, it's a sugar supplement, those tiny packets that you can add to your coffee if you don't want to add sugar to it, but you want it sweet. It's not sweet to Drosophila, because these molecules are artificial agonists. They just happen to activate our receptors and trick them into responding as if there was some sugar present, even though these are not sugary molecules. But receptors in a Drosophila are just slightly different, and they're just different enough that same molecules don't work on them. But that's one part of the story. The fact that Drosophila simply doesn't have any receptors for umami means that they're not carnivorous, means that they don't eat meat and have no sense in detecting it. And the opposite is kind of true for cats, for example, who don't typically eat fruits. Cats are carnivorous, and they are more carnivorous than even dogs and wolves, in the sense that they are obligate carnivores. They only eat mice and birds and stuff like that. So dogs and wolves can taste sugar, but cats cannot. What does it mean in terms of evolution? It would be jumping ahead a little bit, but cats and dogs probably have a common ancestor. So there was an animal at some point that was like this proto-carnivorous animal, but it probably wasn't a specialized carnivorous animal. It was just some random mammal. And probably this mammal had a receptor for sweet because it ate everything. But then this proto-animal had so many kids, the line started to diverge, and some of them started to become dogs and some started to become bears, minks and cats. And in those who continued to eat berries and other fruits, if they would have lost an ability to taste sweet, they would have lost a little bit of useful behaviors, because they wouldn't eat berries, they wouldn't find berries attractive, and suddenly they would be at a disadvantage with other animals of the same species who would continue eating berries. So bears obviously can taste sweet because it's important to them. Sweet things are a part of their diet. While for cats who switched entirely to mice and birds, the situation was different. If one cat at some point, the proto-cat as well, lost the ability to taste sweet, would it have noticed? No! At this point it doesn't eat any sweets ever. And so this mutation is totally relevant for it. It would lose the ability to sense sweetness, and that's okay. The cats don't care. And see, there is some rhyme and reason here, because otters, for example, also don't taste sweets. Because what do otters eat? Fish and stuff, right? Sea products? I'm not quite sure. But definitely not berries. Okay, next, human receptors. There are these lovely, wonderful pictures of human tongues in every textbook about physiology that look so gross. And there are several types of sensory thingies, and they have sensory cells inside. Each human taste bud has 100 receptor cells. All receptor types are mixed, so that's where this idea of different parts of the tongue being responsible for different tastes is wrong. But the mix itself is slightly different, so some parts will have more receptors for sourness and fewer receptors for bitterness and so on. So that's where this idea of a map, of a taste map, is not that stupid. The truth is somewhere in between. Next, where do the projections go? And again, we haven't actually talked about the morphology of the brain yet. We'll discuss it later. But for gustation, the pathway is similar to that for touch. We have sensory neurons that communicate to the medulla. Again, that's in the hindbrain, in the brain stem over here, that communicates to another part of the brain called the thalamus, which communicates, among other things, to our cortex. And that's how we know we have conscious access to the perception of taste. That's what we have for taste: sensory receptors, medulla, thalamus, cortex. That's what we have for touch: receptors, medulla, thalamus, cortex, kind of remember the idea that there are several stops, because in a while we will visit a type of sensory modality that doesn't have that many stops, and we'll have a little story about it. But other than that, in this course, we can afford to park it, although you would probably learn more if you studied human anatomy and courses like that, or if you're preparing for medical school, you would actually learn this stuff and try to memorize it. Next, remember this discussion about champions, about champion species, that for every sense, for every behavior or phenomenon, there is probably a species that invested so heavily in it evolutionary that you can use it for research if you wanted to research this thing. So I'm not sure these animals are actually used for research that much, but are there any gustatory geniuses? Like, there are birds, for example, that can see very well, or you probably know animals that can hear very well. Are there animals that can taste very well, that can taste so much that it becomes an advantage over other species they live with? Let's figure out, are there animals that can taste so good that the taste receptors are not even in their mouth but all over their body? I mean, Drosophila are kind of like that, but are there any ones that push the envelope even further? And what can we guess about these animals? Where do they live? What do they do? What is their ecology? What is their environment, and what are their behaviors, and how would they look like? And also, how would their brain look like? Which is an interesting topic that we haven't talked yet about. But if an animal uses a certain sense a lot, how would you expect its brain to look like? In the previous lecture, we discussed how for touch we have areas in the brain that are responsible for processing touch, and how information from different parts of our body would be probably processed by different parts of our brain, which kind of suggests that just looking at the brain, you can make some guesses about where the hardware is invested, right? If lots of computations are happening, probably you will have more neurons dedicated to it, and the other way around. If you don't use a certain taste, the part of the brain that's responsible for it will probably be tiny, teeny tiny. So think about these two questions, pause the video, then check out the answer. One gustatory genius apparently is a goldfish. Who would have thought? Goldfish, people keep goldfishes in tanks and they look at them, and then there are all these strange legend about goldfish memory that is supposed to be particularly bad. It's not, it's totally an urban legend. They have normal memory like most fishes, which is not excellent but also pretty good. But the interesting thing is that apparently the entire body of a goldfish is covered with taste receptors. So when goldfish is swimming in the water, it tastes this water all the time. That's weird, and it is very invested in this taste perception. And if you look at its brain, it has a part that most fishes and all other animals don't have, called a vagal lobe. So look at that. And again, I'm sorry that you have to engage with a picture of a brain before we actually discuss how brains look like. But look, this part is rostral, so it's looking towards the head. This part is caudal, so it's looking towards the tail. This is actually a spinal cord, so this part is connected to the tube that's running along the fish's body. And different parts of the fish brain are responsible for different activities and different types of calculations, computations. This part, for example, is mostly about vision. So just looking at the size of this part, we can figure out how important vision is to this fish, and it's fairly important. But look at this part. This entire thing is about taste. And in a normal brain, this part would have been much slimmer. This is the medulla, and it's typically elongated. But here we have this blob of matter sitting on top of it that is entirely dedicated to processing taste. Somehow goldfishes are taste geniuses, and we totally don't realize that, and we miss that. It doesn't mean that you need to add salt and sugar to your goldfish tank. Please don't do that. That would be cruel. But in their natural habitat, they use taste to find food or to do something else. I'm not sure. A much neater story and easier to relate to is with another gustatory genius, the catfish. The catfish has these long whisker-like protrusions, tentacles, and these tentacles are all packed with taste receptors. And with catfish, we actually know exactly how they live, right? They live on the bottom of a lake or a river. They live in silt. They live in murky waters where you cannot rely on your eyes. You cannot see there, but there is lots of stuff there to eat. So what they do, they go through this silt and they put their tentacles in it and they taste everything. So like, remember there was this star-nosed mole that used to touch everything? Catfishes taste everything. They taste all the stuff around them before it comes to their mouth. They taste things while they're outside. And if any of these tentacles touches everything tasty, like another animal, then they turn and eat it. And the little funny thing is that they also have a part in their brain that's unusually large, that's dedicated to taste, but because they taste not with their body, but with these extensions, it's actually the different part of their brain that evolutionarily became larger. So while normal fishes have this part relatively slim, both goldfishes and catfishes have some special structures in their brain that are dedicated to gustation, but these are different structures. So it's like there is a theme in evolution that these two fishes discovered independently, and they came up with different solutions for the same problem. And if you look at the brains, you can see that they work slightly differently, but at the same time, there are some parallels. So it's like birds that can fly, but also bats that can fly, that developed wings independently, and the wings are slightly different but serve the same function. Next, now we can talk about olfaction. We're done with gustation, we're done with taste, and we can talk about olfaction, which is the sense of smell, of course. Let's start with receptors. Where are the olfactory receptors? In humans, olfactory receptors sit in the olfactory epithelium. And this is this funny part that's inside our nose. Remember this horrifying picture of why coronal projections are not particularly cute? Because if you make a coronal projection, it cuts through all these special organs that we have inside our head that make dry air a little bit more moist and cold air a little bit more warm, so that it wouldn't hurt our lungs. So there, in this organ, we also have the part that we use to smell things. And that's why we cannot smell very well when we have a cold. When we have a cold and we cannot breathe through our nose, this whole part is swollen, so the air cannot go through and cannot activate the olfactory epithelium. And the situation here is kind of funny because with an eye, for example, you can have an eye hidden inside your body as long as there is some transparent window in your body. The light can get in and you will detect it. You don't have to have the eye exposed to the elements. But with olfaction, our nervous endings have to be sticking out in the environment to a degree. And so this is the compromise. We have a part of our brain called the olfactory bulb growing against the part of the skull that faces this nose structure. Here is the olfactory bulb, and see, it lies on a part of the skull that's called a cribriform plate. And this part has lots of little holes. And there are neurons called the olfactory receptor cells that are sitting in the epithelium and are sending their axons through these holes to connect to neurons in the olfactory bulb. These olfactory receptor cells are therefore on the border between our body and the elements of the environment. They are in the mucus, in this mucous membrane. They have to be moist all the time, otherwise they will die. And they're hidden inside our nose, so to say. But at the same time, there is air, angry, dry, hot or cold or dusty air that's going near them, and that's why they die all the time and new ones are born. So that's one of the few places in our body where new neurons are generated all the time, just because they are kind of vulnerable and discardable. We just replace them all the time. Good, so these are the olfactory receptors. Before we talk about what happens next to the signal, let me ask you the following question. Why do we have two nostrils? And this is a nice question where you can try to answer. So pause the video, think about it. Why do we have two nostrils? What is your guess? And if you have a guess, how would you test that your guess is right? Imagine the experiment that one could run in order to test this guess of yours. Pause the video, compare your answer to the answer in the video. Alright, why do we have two ears? to hear in stereo, right? To compare what we hear with the right ear with what we hear with the left one. And we'll talk a lot about that. Why do we have two eyes? Same thing, kind of. We compare what we see with both eyes, and we'll talk about that as well. So two nostrils, can it help somehow to figure out where the smell is coming from? And the nostrils are so close to each other, and we humans so don't use our smell to locate prey at this point that it may sound weird. Is it even true? Can it be true? But it turns out that it seems to be true because people have studied that. The way they studied that, they hired lots of undergraduates, college undergraduates, and paid them for crawling on the grass, and there was a trail of some smell on the grass, and they were supposed to find where the trail leads to. And some of them had both nostrils open, but some of them had one of these nostrils plugged in with lots of cotton. And what researchers had found is that, first of all, humans can follow a trail. It takes them a while and it looks ridiculous, but they totally can. And second, if you have both nostrils open, you're much better at locating the source of the smell, because it looks like, even though we don't think about that actively, our brain does actually compare what our left nostril smells to what our right nostril smells, and we can figure out whether the smell is coming from, which is weird but seems to be true and was checked in an experiment. Now let's talk about olfaction, the sense of smell in different animals. What animals are good with smelling? You probably know some, like dogs are pretty good, right? But let's go a little bit more general. Let's think of several different points and try to build a nice theory of why certain animals are better at smelling and why certain animals are worse. So here are some points to consider. First, smells are about detecting chemicals. The way our body detects chemicals is by using a protein that binds this chemical. So this is a receptor, but in the sense of a protein that actually attaches to a chemical or allows the chemical to get attached to it and responds to this change in some biochemical way. So if each receptor that can detect a chemical is a protein, what does it mean? Where do proteins come from? Do you remember the basic idea of where the proteins come from? Here's the basic idea. Proteins come from genes, right? If you have a protein, it means somewhere in your DNA there is a gene that encodes this protein, and there is some RNA in the middle, so there is this whole process of how proteins are actually made. But what's important to us, that each protein is encoded by a separate gene. A gene is a part of a stretch of DNA that describes in some code what amino acids should go in this protein, and it has a start and it has an end. So it's a discrete structure that is inherited, and it can be broken or it can be fixed, it can mutate a little bit, and so on. Which is a point number two. Mutations in the DNA happen all the time. Sometimes these mutations are really bad. So bad that the mutation kills the organism. If this protein is important for the development of the body and the protein is broken, then the body doesn't develop. Sometimes they actually confer some interesting property that previous generations didn't have, and that's cool. That happens very rarely, as you can guess. So in most cases, something is broken. Most mutations break things rather than building things. But then the amount of damage can be different. If you're a cat and you lost your ability to feel sweetness, who cares? You don't care, definitely. But if you're a cat and you lost your ability to smell mice, for example, that's gonna be a problem. So from lethal, that kills you, to completely neutral, when nobody cares, with everything in between. If we think real deep about olfaction, about ability of different animals to perceive different smells, what can we tell? What predictions can we make about animals that have few olfactory receptors, but all of them are functional, have lots of olfactory receptors, all of them functional, or they have lots of olfactory receptors and most of them pseudogenes? What does it mean? Olfactory receptors, those proteins that detect different chemicals, all belong to a certain family. Genes for olfactory receptors are all kind of alike to each other, because originally probably there was only one protein who was linking to only one chemical, but then this gene was duplicated. That's kind of more advanced stuff about how evolution happens, but you can get the main idea. There was a gene that described how to smell one chemical. Then at some point by mistake, there were two copies of this gene, but they were saying the same thing. Then one of them got mutated, and now it was able to smell another chemical. And so now you would have an animal who can smell two different chemicals. And in this way, you can get more and more olfactory receptors over the course of evolution, or some of them can break down and stop being functional olfactory receptors. So if we look at different species, some of them have lots of genes for smelling stuff, and all of them are functional. And some of them have lots of genes that look like genes that may help an animal to smell stuff, but most of them are broken. And you know that a protein is broken, for example, if it has a command, the protein is over in the middle of it, because there are commands for different amino acids, but there are also commands for stopping the production. So if by mistake you have a command done, the protein is over, in the middle of a protein, this protein clearly won't work. Even if the body tries to synthesize it, it will just synthesize garbage that will go into garbage, which is really what happens. And so you can look at different animals and look at the olfactory receptors, look at the genes for olfactory receptors, look at the genomes. You can analyze DNA these days, and you would see that some animals have fewer of them and all of them work. Some animals have lots of them and all of them work. Some animals have lots of garbage, like genes that look like they should be helpful but are all actually broken. What can you tell about these three scenarios? And once you think about it, the follow-up question, if you guess the genetic landscape for bees, dogs, and humans, one of them belongs to one category, another to a second category, and the third to the third, but which one is which? Try to figure it out. Think of that. Pause the video. That's a tough question, so you may want to think about it a little bit longer. Think about evolution. Think about the history of evolution. Think about what it means to have lots of functional receptors and what it means to have lots of broken ones. Then continue the video and compare your answer to mine. Let's check the answers. Here's the answer. I looked up the number of genes for olfactory receptors in different species, differently for functional ones and dysfunctional ones. In this table, the first row is the total number, and the second row is the number of olfactory receptor genes that are functional. And look, we have lots of these genes, right? 1% of the mammalian genome approximately are genes for olfactory receptors, which is weird in a way because olfaction doesn't feel like a big part of our lives. 1% is actually a lot. But the reason for that is that you need a new gene for every smell that you're trying to detect, because every molecule that you're trying to identify has a unique structure. So you have to have a protein of a unique structure to bind to this molecule, but not to other molecules. The only way to perform chemical analysis in your nose on the go is to have lots of very specialized machines where each machine catches molecules of a particular shape. So let's look at dogs, bees, and humans. If we look at a dog, dogs are good with smell, right? We know that. And they use smell a lot, which means that it is important to them. So what would it mean probably for an animal? It would mean that it has to have a rich olfactory life, so to say. Dogs are olfactory animals. Smells mean a lot to them. So look at that. They have lots of receptors overall, which means the richness of experience, and most of them are real genes. Most of them are functional. Some of them are dysfunctional, some of them are lost, some of them are now just these signs of the former glory in the genome, scattered over the genome, useless but too hard to clean out, because there is no process in which evolution would deliberately clean out garbage from the genome. It just sits there until it's lost, and it takes much longer to lose something than to acquire it in this case, because nobody goes through the DNA and checks whether every particular part of the DNA makes sense. That's the difference between like our nice Archangel Gabriel stories and actual evolution. This broken gene will sit there for millennia and just gradually degrade, because every now and then, while copying, it would acquire yet another mutation. But because at this point it's dysfunctional, nobody cares anymore and nobody checks. So these are dogs. What other animals are like dogs? Look at rats. You probably haven't thought about a rat as a super dog, but from the olfaction point of view, rats are totally super dogs. Look, they have 50% more genes for smelling and almost twice, slightly less, but a lot of those are functional. So rats are much better than dogs in terms of their smell. Why is it so? Can you figure it out evolutionary? Kind of, right? Ecologically almost. Dogs don't happen to live in underground holes. Dogs mostly hunt during the day because like wild dogs are called wolves. And wolves can hunt at night, I guess, or at dusk, but mostly they're active during the day, aren't they? I'm actually not sure. Anyways, they rely on their eyes a lot as well and on their ears, while rats are these night creatures and also underground creatures, so smelling is much more important for them than sight. Let's look at a bee now. What do we see with a bee? The number is very small. Well, it's still pretty huge if you think of it. It's still at least 160 different smells that a bee can smell, and almost all of them are functional. What does that mean? Essentially, it means that if there is a mutant bee that lost an ability to sense one of those smells, this bee will be at a great disadvantage. The set of smells that they have is pretty limited, but all of them are important. They're not sensing stuff just for the sake of it, just because it may be useful at some point, which is the case for rats and dogs that can learn a new smell they have never smelled before that is not biologically relevant, because who knows, it may be useful. Like, they can use dogs to smell explosives. Like, why would a dog be evolutionarily able to smell explosives? It just has a landscape that's so rich that it can even learn arbitrary smells. Not the case for bees. If you look at fruit flies, not the case for fruit flies. Fruit flies are so conservative, look at that. Almost every single gene, with the exception of one, is functional, which means that each of these smells is important for a fruit fly, for its survival. And in case of bees, you can guess that maybe some of them are related to flowers, maybe some of them are related to predators. I'm not sure what bees are smelling, but they probably smell flowers. Why do flowers smell? In order to attract bees. So there is a tight collaboration here, and a break of trust on the side of bees would lead to a big disadvantage. Now let's look at humans. In humans, most olfactory genes are pseudogenes. And the same is true for chimps. Look at that. So we have relatively a lot genes overall. So it looks like we should be able to smell pretty well, but then it turns out that most of them, more than the half of them, are broken. Signs of the former glory. It's like ruins of castles. It's like arriving at a town and seeing that most buildings there are in ruins and nobody lives there, and the roof collapsed and there are no windows and the vines are taking it over. What does it mean? It means that in the past this town was rich and now it is in disrepair. Nobody cares about it. And if we translate this sentence in the world of ecology and animal neuroscience and animal evolution, it kind of means that our ancestors cared about olfaction and we don't. And that's kind of what happens, because both we and chimps are diurnal species, and most of our ancestors probably were active during the day. So we rely on our sight a lot, we see a lot, and we use our eyes a lot to understand what's going on. And we are not particularly good hunters in terms of hunting by smell. We use different ways to hunt, different styles. Even when we hunt, we don't exactly chase our prey by smelling the ground. And that means that olfaction is not as important to us as to a rat or even a dog. But our ancestors long ago were probably kind of closer to rats in terms of their ecology. And so what happens is that we bear their heritage, but we don't care about it. We live in this abandoned town of smelling. And that's not to say that our sense of smell is particularly poor. We are better than, say, bees or fish or chicken. Compare us to chicken. Well, actually, we're about the same level as chicken. If you look at the number of active genes, okay, take my words back. We and chicken are kind of similar in terms of at least the richness of our olfactory experiences. But still, it's not that bad. It's only about 2 times worse than a dog's. And again, not in terms of sensitivity, but in terms of richness. But we used to be so much better. Oh well. Next, olfactory bulbs. We'll talk a little bit more about that when we talk about brain structure in general, but there are these special areas in the brain that are about smell. They're called olfactory bulbs and they are sitting in the front of the brain. Now, human brain is so large that they are no longer the front, they're kind of on the ventral side, but still you can see the same theme. And the idea is that they're protruding from the brain, which totally makes sense if you think of the shape of the brain inside the head. This one on the left is a crocodile or alligator. So if you think of the shape of the head of an alligator, it's kind of like that, right? And the brain is somewhere over here, but the smelling happens somewhere over here. So it only makes sense that the brain kind of reaches out towards the smelling part. That's why olfactory bulbs in most animals are extended up front, and if the animal has a longer snout, then the olfactory bulbs are probably extended even further, because this part should be placed kind of against the olfactory epithelium. And olfactory bulbs are very interesting. They have fancy structure. They're made up of these glomeruli, which are little parts, and it looks like every glomerulus is responsible for a smell. Now, this is complicated and this is an active area of research, so we won't dwell too much on it, but just as a foreshadowing for some other courses, not even for this course, think about that. In the olfactory epithelium, all olfactory receptors are kind of scattered, there is no order, but then they grow and they connect to different glomeruli, these groups of cells in the olfactory bulb, and they synthesize the experience of smelling. That's what's probably happening. And it's interesting because these scattered neurons send the axons, and from the entirety of the olfactory part of your nose, they reach this one glomerulus in the olfactory bulb. So let's look at red cells. This red cell and this red cell that may be on the opposite part of your nose are sending axons that get together like on a highway and then connect to one part in the olfactory bulb. This process is called axon guidance. It's about axons finding way in the body, because another thing that I've mentioned, olfactory receptors recycle all the time because they die a lot, because they're exposed to the elements. So this process happens again and again and again in our body, which makes it a very interesting model for studying how brains develop to begin with. How do little cells know where to grow? How they decide where to connect to? Because if they connect incorrectly, you won't be able to smell correctly. You will mistake one smell for another. So that's interesting. Another thing is that it looks like glomeruli that are nearby encode similar smells, like flowery smells in one corner of the olfactory bulb, or like meaty smells in the other, pungent and dangerous smells in the other. And we still don't know how it works, how it happens, how can you arrange them in a meaningful pattern before you know, you personally, as a developing organism, before you know what the smells mean. How does it work, especially considering that the chemical structure of different smelly molecules is completely arbitrary? Well, not completely, but pretty arbitrary. It's pretty hard to guess from a shape of a molecule, at least to a naive person, how it will smell like. There are apparently some rhymes and reasons, and chemists know them, but it's not a trivial topic. So people study that a lot, and it's still an active area of research, and we don't know much about that. Or rather, we know a lot, but there is no simple way to describe that. If you look at the circuitry inside one glomerulus, it's even more complicated. And mostly I'm showing these pictures to show how pretty and beautiful they are. There are special cells called mitral cells that have this funny shape with long arms kind of extended. And there are some other cells called tuft cells with a similar but slightly different funny shape. So it's so interesting and so inspiring, and it almost feels like one could understand it, but up to now we don't quite know how it works. There are lots of theories that try to explain different parts of this picture, but no clear story that I could have told you. One thing that I find really entertaining is the types of coding that are used here. If I asked you what coding, remember those codes like rate code and temporal code and population code and so on, how do you encode smell with spikes? So that's weird. At the level of olfactory epithelium, we essentially just by design kind of have a complex population code, because there are lots of olfactory receptors and some of them are active and some of them are not. So somewhere in this picture of which of those are spiking and which of those don't, the smell is encoded, but to classify this pattern of spiking into something meaningful, so to detect what smell it is not a trivial task. within the olfactory bulb, if it's true that every glomerulus encodes a single smell that's synthesized from these elementary reports from different olfactory receptors, if each glomerulus is a smell, then it's almost like a labeled line, except it's not communicated anywhere in the labeled line, so I'm not even sure it's fair to say that. But let's say each glomerulus is the elementary smell. But then the way the olfactory bulb communicates to the brain is using temporal code. And on the surface, this makes no sense at all, because temporal code is about when the spikes happen. And of all senses, smell is probably the one that feels least like a when sense. It's a what sense, right? We don't ask when you smell. We ask what do you smell? Why temporal code is even a thing here? When a smell is present, it's present for seconds or minutes or hours. We actually stop sensing smells when they're present for hours. We kind of get used to them and they stop being reported to the brain. But still, smells linger. Why would you use temporal code to encode them? But for some reason, it's mostly temporal code. And of course, it's not studied in humans because you cannot record from this part of the human brain because it's hard. And yeah, you basically don't do that. But people record from this part of the brain in mice and rats, and this is a temporal code linked to their sniffing. Remember sniffing? There is a process of inhaling and exhaling that increases the amount of interaction between the air and the olfactory epithelium. And because sniffing has this periodic structure, there is at least some temporal structure to the process to which you can link your spiking. And it looks like it's like a Morse code of sort. If it's one smell, neurons are saying pam 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 with every sniff, and if it's another smell, neurons are saying pam 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 with every smell. I mean, I'm making stuff up, but that's the style of it. For some reason, it's temporal code, and if you think long about it, it's kind of mind-blowing, because that's the part of the brain where I personally would have least expected it. But let's go further. Accessory olfactory system, what it is. It turns out that most vertebrates, except humans, have not one but two olfactory systems that are mediated by two different organs, one that we have just described and this another one, and are innervated with different nerves. How come we never heard about them? Because we don't have it. But you can really notice it in snakes and in cats in two different ways. In snakes, that's the main system that they have. And you know how a snake looks like, right? And even a snake from a cartoon or an emoji has its tongue extended quite often, and the end of it has a fork, and the snail would extend its tongue and then bring it back, and extend it again and bring it back. Why is it doing it? And also, why is it forked? It turns out that every time it brings it back into the head, it dutifully puts these two tips of the tongue on the ends of the fork into two special cavities inside, which are called the vomeronasal organ. It puts these tips into these special cavities because that's how it smells. So the tongue is a little bit vaguely moist, and all the molecules in the air got trapped in the moisture in the same way it happens in our epithelium, and then the snake brings the tongue back and puts it into this detection chamber of sorts, and that's how it smells. And why the tongue has a fork? For the same reason for which we have two nostrils, right? As us, snakes smell in stereo. So in snakes, that's the main way of smelling. In cats, it's a secondary way, but still very important. Everything that's related to normal life and to hunting, for finding your way in the forest or in the house, for everything like that, cats use the normal olfactory system that we call normal just because that's the only one we have. But for social interactions and for sexual interactions, cats use the other one. It's also called a vomeronasal organ. There is a second name, Jacobson's organ, and that's where the pheromones are detected. Remember pheromones? Special chemicals that are used for social communication, that are produced by this species for other members of this species in order to communicate something about whether it's male or female, ready to reproduce or not, about whether it's angry or not, about how it feels about its territory, or whatever one member of a species need to communicate to another member of the same species chemically. And you can notice when cats use this organ because access to this organ doesn't go through the nose, but kind of through the mouth. So when cats are trying to use it, they open their mouth in this funny and a little bit scary and weird way. When you see two cats interacting on the street, when they're potentially aggressive to each other, they have this strange snarling smile on their faces, and their mouths are a little bit open, and part of that is an aggression display, but part of that is an attempt to smell each other using this special organ. And humans don't have it. There is a little bit of controversy about that because human embryos seem to have the nerve that would have led to it, but at the same time, if you try to find in a human the genes that supposedly should encode the olfactory receptors for this special secondary organ, they are all pseudogenes. So they are formally there, but they are non-functional, dysfunctional, like as we've seen in the previous picture, which kind of suggests that it's not functional in humans. And it looks like humans don't actually have pheromones. So every time a perfume company is claiming that it adds pheromones to its perfume, that's basically a lie. They're just trying to sell more stuff because it kind of sounds crazy. Pheromones, mind control. Humans don't actually have it. Sorry. However, there is an interesting story about the sense of smell still. Let's remember how gustation worked and how touch worked in terms of its connections to the brain. There was a sensory neuron that projected to medulla, that projected to thalamus, that projected to cortex. What's the deal for smell? We have an olfactory bulb that projects directly to the cortex. It projects directly to the part that we use to think. And in a way, it seems to be an evolutionary peculiarity, because cortex originally was just this frontal part of the brain that was mostly dealing with smells, and then it started to grow. This is an oversimplification, of course, but we now use this part of the brain to think. It has become huge, and that's the first and almost only thing you see if you open a human head and peek inside. But originally it had something to do with smell, and it still has the connections. Smell is reporting directly to the cortex, and it doesn't make these synaptic stops in the depth of the brain. And it may actually mean nothing. It may be just a funny arrangement of connections, like who cares, right? But at the same time, it looks like we are not always consciously aware of weak smells, even when they affect our mood. Olfaction seems to have a slightly privileged position among other senses. We form associations with smells much better. Most people are not able to imagine smells, but at the same time, we remember smells, or rather not even consciously remember them, but remember emotions related to smells really strongly. If you listen to 21 Pilots, they have this wonderful song that mentions olfaction, the smell of a candle that smells of your childhood, which is such a nice neuroscience type lyrics. I really love it. The idea that certain smells can have this deep importance to us that's almost weird. And of course, there is classic literature about that as well. So despite the fact that we don't have pheromones, smell seems rather special. And interestingly, anosmia, inability to smell, leads to depression more often than, for example, blindness. Even though a person who lost the ability to see or the ability to hear in our modern life is much more challenged, and it's harder to live without sight or without hearing than without smell. Maybe that's because most of what we think as taste is actually smell, which is something you notice when you have a flu and you cannot smell anything, and so all food tastes bland, because most what we perceive as taste is actually smell. As we've just discussed earlier, we have only 5 different taste receptors, so it's like our gustatory landscape is really, really poor. All the variety of food is about texture and smell of it. Maybe that contributes to this increased rate of depression, I'm not sure. But smell is interesting. Next, now we have a really brief foray into various weird senses. And they're only weird to humans because humans are just one species. They're totally not weird to other species. So maybe I shouldn't have used this name at all. Maybe I should have called it non-human senses. Lateral line. We talked about the lateral line, right? It's hard to even quantify it. Is it touch or is it something else? But fish have this special line along their body, and frogs have it too, and salamanders, where there are special canals that are kind of hidden inside the body but have numerous connections with the outside, and they have hair cells. Hair cells are not like hairs. They are what we use to hear, and we'll talk more about that next week when we start talking about hearing. But in this case, they don't detect sound. They detect slower motion of fluid, of water, through this canal, which means that a fish is always acutely aware of how the water is flowing along its body. And even if a fish approaches a wall or another fish, slight changes in the way water circulates around it allows it to feel it long before it actually touches it. So ecologically, it's like distant touch. Physiologically, it's close to our hearing. And overall, it's just something we cannot experience because we don't have it. The closest we can get to this feeling is if you have hairy arms, like I have reasonably hairy arms, then you can feel wind, because wind changes the position of your hairs and you just feel it. So imagine that fish can feel water like that, only much better, because their brain works with this information all the time, partly innately perhaps, but partly through development. They learn how it feels to swim past another fish or past a stone or a piece of seaweed. And the lateral line is pretty noticeable in a fish. If you look at these lines that run along the fish, it's not the spinal cord, obviously. Spinal cord is somewhere inside. It's the lateral line. These stitches are where the entry into this canal is. And then there are even more on the head that are not particularly visible here, but in some fish species they're pretty obvious. Next, electroreception. Many animals can do something with electricity. There are 4 types of interactions you can have with electricity. The first option is to have no electroreception, in which case you cannot feel electricity. And humans are there. We cannot feel electricity. If somewhere near us is another animal that is contracting its muscles and action potentials are generated, we would not know because we cannot feel it. But many animals can. And it's kind of interesting if you think of that, because electricity is so important in our culture and in our civilization, and yet we don't have an intuition for it because we cannot feel it. Remember this very first lecture that I gave about ions and action potentials and charges and how ions are charged? And I tried to convince you that that's okay if it feels weird to you, because unlike mass that we can feel directly if we try to lift something, or unlike color that we can see with our eyes, we cannot see charges, we cannot perceive them, we don't have any intimate relations with them in our childhood. And the situation is very different for many other species actually. So if electric fishes, about which I'll talk in a second, if electric fishes would build a civilization, for them electricity wouldn't be a problem. In the first lecture of a fish course, a teacher fish would talk to pupil fishes, no electricity, and the pupil fishes would say, yeah, of course, we are not babies, come on, we're using electricity daily. We know how it feels like. Because many animals, as it turns out, fall in this second category called passive electroreception. Passive electroreception means that you don't generate strong electric fields yourself, you don't generate them for any special purpose, but you feel electric field generated by other animals. Because we all generate electric fields all the time. Why? Because muscles use action potentials. And these action potentials are fairly strong. And the fibers in the muscle are all oriented in the same direction because the muscle needs to contract. So these action potentials are somewhat aligned, which means that when an animal slithers, crawls in the silt somewhere at the bottom of a lake, it always generates weak electric fields and weak currents around it. And you cannot see in the silt, you cannot smell very well in the silt because all of it smells. You cannot hear in the silt. Well, you can, but maybe not too well. What can you do? You can taste with barbs, or you can try to catch these tasty electric fields that will lead you to food. So many animals have passive electroreception, including sharks. Among mammals, platypus and echidna have them. Dolphins also can do it apparently, which was news to me. And potentially more animals may have it, and maybe we just haven't noticed it yet because we just haven't tested. For example, Wikipedia claims that frogs and other amphibians don't have electroreception, but if you look on Google Scholar, at least some frogs and amphibians seem to have it. So I guess you can go and edit Wikipedia if you want to. It looks that many animals use it at least to some extent, because actually it's not that hard if you live in the water. But then some other animals use electricity even more actively. And there are 2 options here, weakly electric and strongly electric. Strongly electric fishes kill with electricity, or maybe not kill but stun. They create discharges that are so strong that they paralyze the prey so it cannot escape anymore and you can eat it safely. The most famous of those is of course the electric eel. That is not actually an eel. You can find some nice pictures of it online where people have to wear rubber gloves to manipulate it, otherwise you will be shocked. But then there are also weakly electric fish, and weakly electric fish use electricity in order to sense the environment and also for communication. So here are some examples. Sharks don't have active electroreception. They don't send signals into the void in order to hear the responses, but they have passive electroreception, which means that even without eyes, they would be able to feel the electric fields generated by muscles of another animal. Where are these electroreceptors? They are on this very picture. Let's find them. Clearly not the eyes, clearly not the teeth, clearly not the nostril. What then? These guys, all these tiny thingies, and these guys as well, are organs of electroreception. They're called ampulla of Lorenzini. People know how they work roughly. Sharks have lots of them on their noses, on their snouts, as you can see. And have you ever wondered why hammerheads have hammerheads? Why would you have a head that's so weirdly shaped? That's in order to improve stereo perception of something, obviously. And they seem to have a very good sense of smell, but they also have electroreception, and they can use this entire head as a detector of stuff. But then there are lots of tiny fishes that use active electroreception in order to find prey. They send these electric waves into the environment, and then they listen using special organs to the response. If they are close to a stone, and stone, unlike water, cannot conduct electricity, they would know it because the electric field would have a different shape. And if they're close to something that conducts electricity better than the water, like a worm, they will know it as well. And they're so good at that they use it all the time. So they have to have some mechanisms in order not to mess with each other electric sensation. And they have a whole politeness about that, a whole ritual called a jamming avoidance response. If two fishes meet and they use the same frequency by default because they belong to the same species, the fish that's more respected, that's higher on the social rank, will go higher, will increase its frequency. They have to diverge their frequencies because they're sensing the environment, but they don't want to jam each other. They have to change the frequencies at which they operate. And who goes higher, who increases its frequency and who lowers this frequency, depends on the social status. Isn't it cool? The interesting thing about jamming avoidance response is that it's easy to study in a lab. You just put two fish in a tank and then you put two electrodes and you connect these electrodes to a speaker. And because the fishes already generate electricity, you just amplify it a little bit and you hear whoo, and then you put another fish and instead of one note you have two notes. I've never done that. I always wanted to do that, but I'm too lazy, and also these fish are kind of tricky and hard to maintain, and they're mostly harvested from the fields, which is apparently not a sustainable practice. So while they're pretty cheap, there are some ethical considerations here. So I really wanted to make sure that I can give them good life before I purchase them, and I never managed to convince myself that I can give them a good life. But potentially it's really interesting and easy to do apparently. Here is another fish that also uses electrosensations called an elephant nose fish, and unlike fishes in the previous picture, that always produces a constant waveform that if you amplify it and listen to it, sounds like whoop. This one uses separate discharges. So again, if you put electrodes connected to an amplifier and listen to response, it's like separate clicks, like chick, 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 chick. They use these discrete signals in order to sense the environment. And apparently these fishes are ridiculously smart, and apparently they can communicate in almost something like a Morse code, like they can use these clicks, these electric clicks that of course cannot be perceived by us without a special amplifier, to communicate messages to each other, which sounds really weird considering how small they are. But also if you look, they have an unusually large brain and of unique structure. There is a strong suspicion that these fishes are much smarter than many of its relatives. And again, I really thought that I want them and I would listen to their clicking and understand their language at some point, but I could never convince myself that I would give them a good life, because they are kind of tricky and require lots of care. Keep an eye on those fishes. They may be smarter than we thought. Alright, let's talk about other types of senses that humans cannot easily relate to. Spiders can hunt using 3D hearing. What is 3D hearing? If you're a spider, you have 8 legs. These legs are standing on the ground. It turns out that sound propagates much better in the ground, in a solid body. Moreover, in the air, there is only one kind of sound. We'll talk about sound next week, but basically something moves back, there is a movement, it changes the pressure of the air, and these differences in pressure propagate like a sound wave. But in the air, the sound is this combination of waves that have a little bit more pressure and a little bit less pressure. That's it. In a solid body, like the ground, you can also have rotational waves, like if everything shakes a little bit or like rotates a little bit. These displacements of particles can also propagate. It doesn't work in the air or in the fluid because rotating the air doesn't do anything to the air nearby. One part of air can rotate easily compared to the other part of air, but that's not true for the solid body. If I have a book and I try to rotate a book, like this part of the book rotating causes this part of the book rotating. That's what I mean. So this rotational wave can propagate with a stone, wood, or ground. If you have 8 legs that are spread on the ground and each leg is enabled with a receptor that can detect vibrations really well, and also there are 3 types of vibrations, you can pretty much understand everything that happens around you. It's like having a super radar. So every spider, like jumping spider or something, standing on the ground is constantly aware about every movement of every tiny particle of grain in the radius of several inches around it, which really helps to hunt. If you think you cannot just run past a spider standing on the ground. It will hear you, and it will find you, and it will eat you. Next, snakes. Also fun. Snakes can sense infrared. They can see the heat of your body. Not all snakes can do that, but pit vipers can. Pit vipers are called pit vipers because they have pits. And here they are. These round structures on its face are not nostrils. This is not what snakes use to smell stuff. Remember, we just discussed how snakes smell stuff. They extend their tongue with a fork, let it catch molecules, get it inside the mouth, and put them into the vomeronasal organ in order to analyze that. So these are not nostrils. These are pits to detect heat from somebody who can be eaten. And the way they work is like primitive eyes, in the sense that there is a hole, there is no pupil, there is no lens, there is just a hole, and then there is a sensitive surface on the other side of the pit that almost works like a retina in the eye. And if a prey is down, then the part of the pit that is up that will be activated. And the other way around, if the prey is up, then the lower part of the pit will be activated. So you can kind of figure out where the infrared radiation comes from. And infrared radiation is heat radiation, is something that we cannot see, but every warm body emits it. And the other way around, every cold body on a warm background will have a shadow of sorts. And so a pit viper can detect mice like that. They can use this sensation to catch warm-blooded food like chipmunks or mice or other rodents and small birds. This picture is obviously from a human camera, human infrared camera, so I'm not saying that snake sees exactly that, but that gives you an idea of how, at least in theory, a mouse can look like for an infrared seeing animal. And again, these are not eyes. This stuff inside is actually a very sensitive skin. So the types of receptors that are involved are the same types that we have in our skin. Snakes just have more of those, and they are arranged in this peculiar manner that makes it possible for them to know exactly where the hot little mouse is. But if you think of it, we can here for a change actually relate to it. If you've ever been around a kettle or around a fire is probably a better example. You know where the fire is even with your eyes closed, because the back of our hands and the skin on our face are pretty sensitive to heat. Even if you close your eyes on an autumn day near a bonfire, you will still know where the bonfire is, just because one side of your hand will feel the heat and the other one will feel the cold. If you never tried it, Try it at some point. I mean, be careful with bonfires. But this is something we can actually relate to. Sometimes we can know whether a cup of tea or coffee is hot or not just by standing near it, because our infrared sensation is actually much better than we realize, and I bet if you practice more you can improve it. And the last one in this story, magnetic sensation, and that's the last one just because it's the most bizarre. Of course there are lots of other interesting senses I just didn't have time to cover, but birds have a built-in compass that helps them to know where the North Pole is. Actually, as it turns out, they have two different compasses, one in their eye and another one in their beak, so they even have a backup version for some reason. Well, we know the reason. Many birds migrate. They fly from one continent to another or far away within one continent. And sometimes you can use sun and stars to navigate, but sometimes the sky is overcast and some birds migrate by night, in which case having a compass in your head is really handy. For a while it wasn't clear how they do that, but now we know that there are two compasses. One in the beak uses little pieces of magnetic iron that rotates a little bit depending on where the magnetic fields are and touches some neurons. And then another one is in their eye, in their retinas in fact, and this one is much harder to explain how it works because quantum entanglement is involved. I don't even quite know what it means. You might have heard about quantum entanglement because it's used in sci-fi a lot, but that's some quantum mechanics stuff, people. Pigeons have quantum mechanics in their eyes to know where the North Pole is. Only think of that. And it was known for a while now that when birds are flying above a magnetic anomaly, a place where there is lots of iron ore under the ground and this ore is magnetized, and so all the magnetic fields are screwed up, the birds also get screwed up, and sometimes they even cycle, or at least they get deviated away from their path. So that's why they have compasses in their head. This is the end of the story. Let's enjoy this pigeon for the last time. Thank you, and see you at the next lecture.
Time to Lord of Smell and Taste.